Right, good afternoon, everyone, and thank you so much for joining us today. It's been a while since our last webinar in June, and it was good to have a break, but good to be back with all of you. So thanks for being here this afternoon. Just to mention before we start that if you do need SACNAS points for this presentation, for this webinar, then please put your name in the chat and indicate that so that we can organize that for you. Right, and then it gives me great pleasure to welcome Dr. Kelsey Glennon this afternoon. Kelsey is a colleague of mine and she's doing some exciting work in the Drakensberg, and we're really looking forward to hearing it. So over to you. Thanks, Kelsey. Oh, and then just to mention, we'll have a question session after the presentation. So you'll be able to ask your questions then. Thank you so much. Enjoy the talk. Over to you.
Great. Thanks, Glennon. And thanks for inviting me to give a SAAB webinar and to hear about the journey of flower color evolution that we've been going on this last few years. So much of this work was done by two MSc alumni in the lab, Courtney Gardner and Promisem Taleni, and then a postdoc in the lab, Steph Payne. So I don't really need to go to convince anyone that flowers are an ideal system for studying variation. We have groups, flowers are amazing for this because of their size, shape, and color. So we've started to think more about the variability that we were noticing when we were out in the field. And we were out in the field looking for Rhodohypoxis species, which is part of the star grasses or the Hypoxidaceae. The Hypoxidaceae is an almost cosmopolitan monocot family. There are somewhere around 8, 9, or 10 genera with around 130 species, and this is changing in the last couple years. Interestingly, the center of species diversity is in Southern Africa, particularly in South Africa. The other genera occur in North America, Asia, Australia, and Africa, but there are no natives in Europe. However, if you travel to Europe, particularly the UK or the Netherlands, you can buy a variety of Rhodohypoxis taxa that have been collected from here, bred into specific types of hybrid genera. So they are present in Europe as well as Japan for this reason. So for this talk, I'm going to start out with like a big picture, family level, what are we thinking about flower color variability in the family, and then zooming in on Rhodohypoxis bowerii. My group focuses primarily on Rhodohypoxis and thinking about polyploidy, hybridization, and of course, flower color variability. So we wanted to learn about color origins and transitions across the family. And to do this, we used a phylogeny from Wei et al. In 2012. And we started out by mapping color. We scoured different field guides, descriptions, monographs, even iNaturalist and other online sites where photos of the flowers were taken so that we could identify different colors within the family. Hypoxis is the most well-known genus in the family, and most Hypoxis flowers are yellow. So we were kind of thinking we wouldn't really find too much variability here. However, we did actually find some color variability, and this is highlighted in these 8 red arrows here. And so every one of these arrows highlights a species where there are more than one color of individuals within the species. And this can be populations that are polymorphic or monomorphic populations as well. So it does highlight that particularly red and pink flower color does arise multiple times within the family. However, there is the overall yellow coloration of the majority of taxa. I do want to point out that I have moved the phylogeny very far to the left of the screen just for visual purposes, but the outgroups to hypoxidaceae are known to be either white or green. And this is interesting once we started delving more into our pigment peel data. I also wanted to note that this tree doesn't reflect the true diversity of the group. A PhD student in the lab, Henny Neiman, has taken on this task of building a Southern African hypoxidaceae phylogeny, and he's been also keeping track of some observations that he and Steph have seen in the field over the last couple of years. And so we hope to have a more nuanced and informative picture of this in the next year or two. So when we think about the colors of flowers, we want to know what makes flower color. And this was particularly important when we were thinking about transitions between the polymorphic species and monomorphic species. So what was happening at the biochemical and genetic level. And we know that there's two main routes for making flower color. The first is pigments. And so we were focusing primarily on carotenoids and anthocyanins. And so we know that carotenoids are primarily responsible for the yellow to orange and red colors of both flowers and fruits, and anthocyanins, on the other hand, are more pinks, purpley, reddish colors of both flowers and fruits as well. And anthocyanins are well known for their role in pigmentation, not just of flowers, but also of vegetative tissue, so stems and leaves as well. And anthocyanins are part of a flavonoid group and play a large role in plant physiology. And so they are often generated as a stress response to things like UV, heat, and drought. And so as we were thinking more about the variability that we were observing, we started thinking about environmental variables and what correlations this might have. The other thing that makes flower color is structure, and there's been some really awesome work done by Alan Ellis and Heather Whitney in Poridia capensis. And so if this is not at all part of what we've been thinking about, but I did want to highlight that structure is found in one of our taxa. It's a non-pigment based means for flower color, and it has to do with the epidermal cell shape. And it's also responsible for the iridescence in that beautiful center of the Poridia capensis flowers. So when we started thinking more about what pigments were present in our own species, we followed a petal peel of the epidermis from Ing and Smith. And so we use these epidermal peels of both herbaria and fresh tissue to get an idea of whether or not anthocyanins and carotenoids would be present if they were co-occurring or if they were separately occurring. And it's really neat. You can distinguish between the two pigments in the cells, where carotenoids are found in plastids, or known as chromoplasts, and the anthocyanins are stored in a vacuole. So these are water soluble, and they tend to spread out within the cell, and they present more of a uniform color. And so here, these are 6 examples of what we found when we did our epidermal peels of the petal tissue. So we found evidence for both carotenoids, chlorophyll, anthocyanins, as well as structural color. So kind of moving in a clockwise direction around the slide, we start off with our chlorophyll. And this is in Saniella, one of the white petaled species. So that was really exciting. I did not really expect to see that. But then thinking about the outgroup of the family, it kind of made sense as to why we might see chlorophyll present in the petals. Then we're moving into Poridia capensis, and this is right along the transition between that center, that bright center, into a white petal. And you can see anthocyanins present here, as well as the conical shape of the epidermal tissue. Next, we have Rhodohypoxis bauerii, and our pink flowers are clearly very pink as a consequence of anthocyanins. Then we have another Poridia capensis, a pink petal this time, and you can see the conical shape of the epidermis, as well as the the anthocyanins. And in some of our herbaria tissue, for hypoxis, we found that there was both anthocyanins and carotenoids present. And so it was a bit more difficult to get some really nice images here, but there are definitely anthocyanins and carotenoids present there. And then the top image on the left is carotenoids in their plasmids within the cell. So we found a whole range of different ways to color our flowers. I was then chatting to a colleague who's a biochemist at BITS.
Can just go under the
He
chair,
was
under the table and plug the.
So I was chatting to a colleague who's a biochemist and telling her about what we were finding with the pigment peels, and she's really interested in anthocyanins for her own research. And she was like, why don't you just send me a couple flowers and we will kind of get to the bottom of what anthocyanins are present in your flowers. So I sent her a few flowers along with a hypoxis, because I really wanted to see if there was a difference between pink flowers and yellow flowers, because there is that overlapping between carotenoids and anthocyanins. And Sadhna found that we have evidence for malvidin in our pink flowers. And so we're still working on which carotenoid is most likely responsible for the yellow color, but for now we know that there are carotenoids and our anthocyanins as likely malvidin. This was really exciting because we had thought a lot about the anthocyanin biosynthesis pathway and how we could figure out which genes were controlling the flower color. And so knowing that it's malvidin, that helped us narrow down onto the groups that we likely had present. And so we have evidence that it's primarily from the cyanidin group. And this is where we would focus our next goals in the genetic pathway. So one of the next steps for us is figuring out which of these genes are turned on and turned off. And this is where I start to get really excited about polyploidy, because it seems that maybe polyploidy might be contributing to the variability in flower color. So if we think about it across the family, there's lots of chromosome variability, and maybe this corresponds to whether or not the pathway is turned on or turned off. So for example, in one of the taxa I'll talk about just now, there are different ploidy levels, and there's also no color. So it might be that this duplicate copies of the anthocyanin pathway might be interacting with each other in a negative fashion to shut down the pathway, whereas in diploids, it's wholly intact. So this is something we are going to explore more with, and then look for some signals in the transcriptome that we're waiting for, and we'll see how that goes. The other thing that we were thinking about is UV. So we measured UV on pink and white flowers just to make sure. So we know that other pollination biologists know that pollinators view flowers differently than we do. So we thought maybe there's something that we aren't seeing that might come up, particularly with UV reflectance. However, we didn't really find any evidence for UV reflectance in the flowers. But what it did help us do is distinguish between the dark pink, light pink, and white flowers. So we could say that there are clear ways of quantifying the color intensity for our dark and our light flowers. So once we had a preliminary understanding of flower color variation at a macroevolution scale, we really wanted to focus on color variability within a species. And Rhodohypoxis bowerii is a species that is near and dear to my heart. I love this taxon. The varieties provide such interesting questions about reproductive isolation, polyploidy, hybridization, and then they come up with flower color variability too. So it's really just a phenomenal little species to ask all sorts of questions about plant evolution and speciation. So there's 3 named varieties in Rhodohypoxis bowerii. There's platypetala, which is a white variety. It's predominantly white. You might find a few pale pink individuals occasionally within a population, and it occurs all throughout the Drakensberg. primarily generally lower in the foothills into the KZN Midlands, but does occur at some of the higher altitude areas too. It's also found on dry rocky outcrops. And this is different from Bowerii, which is a very bright pink variety. Bowerii is often found along little like seeps in sandstone areas. So we went to a farm down in the Eastern Cape around Maclear, and there was a gorgeous sandstone outcrop, and my family really wanted to hike out to it. I was a bit tired from having gone up and down Nadir's Neck Pass looking for plants, but they really wanted to hike out, so we did. And lo and behold, there were tons of little Bowerii. So I was very happy to have taken that long hike. And these little flowers were just along the edges of these very damp seeps. It was really nice. When they revised the genus in 1979, they named Rhodioboxus Bowerii var. Confecta. And the name is from the fact that when you look at a population, it looks like confetti sprinkled across the mountaintops with these bright whites and pinks and then these pale pinks. An interesting observation that Hilliard and Burt had made about this variety is that they hypothesized that the flowers turned pink over the flowering season, and that they started out white and then started to go pink. And on the right-hand side of your slide, you'll see these 4 images, and these are all confecta flowers that we collected from one of the populations. And so this observation led us to think more about what drives this observed variation in this taxon. And from here on out, confecta kind of takes center stage into our exploration into flower color. So the obvious question to ask is then what drives this observed variation? And there's 2 main flower color selection agents. First is biotic, and then the second is abiotic. So I give the example of snapdragons here for pollinators maintaining polymorphisms, but there are loads of examples in the literature and all around us. I just really happen to like snapdragons, so I thought I'd showcase them here. On the flip side, there's Linanthus perrier, who is a really nice example of abiotic selection agents, and such as spring precipitation, where you have this intra-population variability. And the thing that I really liked about this study that made me think more about confecta was that there was a change in the color morph frequencies from year to year. So one year there would be more blue morphs, and other years there would be more white morphs. And originally, Wright had written about this group or this taxon and suggested it as an example of genetic drift. And then many years later, Shemsky and Birzadek came along, and they did a very long-term study on Linanthus perrier, measured the floral color frequencies from year to year, measured selection agents, and found that it's actually spring precipitation that affects the color morph frequency. So these studies underscore the importance of thinking about selection agents when trying to explain what's happening with your flower color variability, both within populations and species. And I want to take just a small detour here about what does flower color polymorphism mean? So this was a question when we first started thinking about confecta, it was like, oh, cool, like Bowerii, if you look at the species holistically with 3 varieties, it makes sense that we have a polymorphic species. We have these white Platypetala populations, we have these pink Bowerii populations, and then we have these totally variable confecta populations. And so Narbona et al. In 2018 nicely defined flower color polymorphism as discrete variants within a population in high enough frequency to not be the result of recurrent mutations. And so when we thought about how to subscribe confecta as to whether or not it's flower color polymorphism or just variability, we kept this in mind going forward. And we realized we had a few questions to answer before we could definitively say that this is a great example of flower color polymorphism. So this was really helpful in tying together the literature and Hilliard's observations, and to think more about what polymorphism means and whether it can be applied to confecta. So we took another step back within the three varieties or within the Bowerii species, and thought about broad geographic and environmental effects on flower color variation. And so on the left-hand side here is a plot of the Bowerii populations that are labeled in pink, Platypetala labeled in white, and then Confecta is both half pink and half white. And so Bowerii, you can see it's more in the southern Drakensberg, whereas Platypetala kind of ranges throughout the Drakensberg into the KZN Midlands. And then Confecta is like dotted along these peaks of the Drakensberg. So it's at high altitude populations. And like I said, the microhabitat of the flower colors for each varieties also seem to match up with what we were finding in the broader scope of their varietal distribution. And so we're currently comparing broad scale abiotic variables from populations of different color morphs, and some preliminary data by lab members suggest that there's differences in broad climatic variables like precipitation. And my reading into this is that this is probably a proxy for soil moisture. And that's just thinking about what we've learned over the past few years. And the work that I'll start focusing on now is centered in the Free State at Sentinel Peak. So we spent a few years at Sentinel Peak collecting color morph frequency data. I still go back every year. My goal is to have a long-term data set of flower color frequencies with the relevant environmental variables so that when I am retired, I will have a nice data set to publish. But you gotta have lofty goals. So for this work, we focused on 3 sites at Sentinel Peak. The first was up at the car park. So if you've hiked to the chain ladder up the zigzags, you've seen the abundance of Confecta along the trails. They're pretty much everywhere. Then we found a site of Confecta at the lodge when we were out walking after fieldwork one afternoon. So we decided to add that in as a subpopulation. And then then the same trip, we had stopped just to admire the view coming down off the mountain, and some white little dots caught our eye. So we got out of the truck and took a walk down and found another population of Confecta along the catchment. So across these 3 sites, we measured temperature, humidity, and soil moisture, as well as counted the number of white and pink morphs. So at Sentinel with Confecta, we set out to learn whether or not Hilliard and Burt were right. Do these flowers start off white, change to a light pink, and then change to a dark pink towards the end of the season? And then what was the role of UV and soil moisture in maintaining this variability or triggering this variability within the populations? We also wanted to get an idea of whether or not color morphs were different fitness-wise. So was there a clear difference in pink morphs versus white morphs in terms of their fitness? And so based on previous work, we expected to find that drought or water deficiency was going to be producing anthocyanins in their flowers. So we expected to find pink morphs where there was little water relative to
Friday or tomorrow we have
So one of the things we did was we quantified color changes over the season by counting morphs. We throw down 12 quadrats at our sites, and we count the number of white flowers, pink flowers, the number of senesced flowers. Once November rolls around, the plants they seem to start setting seed more then. We don't usually see it too early in October. So then we started collecting seeds. And then within each of our quadrats or along the transect, we record our temperature and soil moisture. And so this really, you get right down into the flowers that are quite small. And we've really learned a lot by being on the ground with the flowers. One of the things that we've done is in collaboration with Sandy and Nick at Kwakwa, we tagged 40 individuals at Sentinel and photographed them every 3 days over the life of a flower. So we had an idea of how these flowers were changing over time. And then we did it across the field season too. And so on the left-hand side of your screen, you see 3 panels of the same individual at 3 different times. And you see there's not really a clear difference in the flower color. So they're not necessarily progressing from white to pink. So we found that the flowers themselves don't change, but then we have some individuals that produce multiple morphs. And on the right-hand side of your screen in the 4th picture, you'll see there's an individual plant here that first a white flower emerged, and then later on in the season, this nice bright pink flower emerged. So there are some individuals that are doing this, but not very many. So they don't seem to be the majority of individuals that we've observed over our time in the population. And so although we did this for 3 subpopulations, the lodge and the catchment site did not show a marked change in frequency, if any. So for purposes of a clear slide without a lot of graphs, I'm focusing primarily on the car park population. And so the car park population was really key in thinking about color morph changes and variability. And it brought up a lot of questions. So here we have the frequency changes through time for our color morphs. The bottom bars are early in the season. We generally get down around October, around mid-October is what we're starting to fine tune our travels. For a while, we were going pretty much every other week to make sure we didn't miss anything. The mid-season is about peak, so that's around the 3rd week of November. And then late is in the 2nd and 3rd weeks of December. My family is not always too keen that on the 23rd of December, I really needed to get down to Sentinel to count flowers, but there you have it. So we have seen over 3 years, and I haven't added in our 2020 data yet, that shows we have more pink flowers later in the season. Interestingly, in 2018, in the middle of the year, it seemed we had a bit more rain than normal. So this might be a reason why we see this increase in pink flowers. Also, that year, the flowers seem to finish earlier than we expected. So sometimes they go into late December, early Jan, and then sometimes they finish right around Christmas. So something was happening there. But I'm hoping as we collect more data through the years, we'll have a nice evening out of these little differences. So when we were out, we were collecting environmental data. This is Courtney Gardner, who a lot of this is from her MSc. So we use eye buttons to collect temperature and humidity, and then a soil moisture probe to estimate soil moisture in each of the areas. And when we started to pair the frequencies or the percent of pigmented morphs in the population with soil moisture, we found that at the lodge and the catchment, there wasn't really too much of a difference. And so this kind of made sense that there wasn't a real, it didn't really seem that soil moisture was driving any change at these 2 sites. But then when you look at the Sentinel Car Park, which is the top line on your graph, that's the higher altitude, you can see that there is somewhat of a relationship between an increase in pink morphs and an increase in soil moisture. So then we started thinking more about the habitat and what these flowers were experiencing. They're at high altitude with a thin atmosphere, and this seemed that UV might be a contributing factor here. And this also fits in with what we know about the relationship between an increase in UV and an increase in anthocyanins from a physiological perspective in other plants and systems, where they're trying to protect themselves against this increased exposure. And so once Courtney finished her MSc focused primarily in the field, but as most science, there's more questions than answers at the end. And so Promise kind of picked up, and he developed growth chamber experiments where we could test how UV and soil moisture were interacting or working on the flower color emergences. And so we hypothesized that if soil moisture and UV did influence flower color, that we would see an increase in pink morph emergence in UV treatments. And so Promise set up 4 treatments in 4 growth chambers. Luckily, our plants are small, so we can fit all the 4 of the treatments in each of the chambers. And then Promise rotated the plants among the chambers on a weekly basis to make sure there was no chamber effect. And then over 8 weeks, he measured leaf length, counted how many leaves came up, counted how many flowers came up, recorded the colors of the flowers, and then made a note as to whether any seeds were produced. And so our 4 treatments were whether or not UV was present or absent, and then whether or not soil moisture was high or low. So we did a pilot experiment because our soil moisture values in the field ranged quite dramatically from about 2% all the way up to about 40%. But in a 10 centimeter pot, 2% made our plants physiologically inactive, so that they were not happy when they were that dry. So we tried to find what was a happy low soil moisture percent. And so around 10% seemed to make them happy enough, but not get them into the well-watered area. And so Promise had these treatments going, measured the traits, and then we figured out what happened with the UV. And so we found that plants in the UV treatments where they were exposed to UV produced more leaves and longer leaves, as well as more flowers and more pink flowers. So this, unfortunately, it wasn't significant, the difference between the UV and the non-UV treatments for the percent pigmented flowers. But in the lower right-hand side of your screen, you can see that there is a trend between the bar graphs. And so the white and the light gray are where UV is absent. And then the light gray, the slightly darker gray and the dark gray is where there is UV applied to these plants. And so there tended to be more growth under UV conditions. And so when we were thinking about what we've seen in the field, what happened in the growth chambers, to kind of get an idea of what's happening, we found that there was an increase in pink flower production at Sentinel, and this likely corresponds to the increase in UV exposure. And when we started thinking about all the pieces of the puzzle with anthocyanins and the flavonoid base of anthocyanins, absorbing UV, that's a high altitude, thin atmosphere environment, solar radiation is strong. So it seemed plausible that plants may be responding to these environmental cues, such as UV, and working towards increasing their pigmentation, not only in their flowers, but also in their peduncles. So the stem under the flower also had a pink or reddish tinge to it towards the end of the flowering season. So having to get down on your hands and knees to count the flowers gave you an opportunity to observe some other traits that we would not have normally seen. And so this might be to prevent UV damage. This also corresponds to the larger geographic expanse. So the pink varieties are all, the pink variety Bowerii is also generally at a higher altitude and more likely to bear stronger UV exposure than the lower altitudes with the Platypetala populations. This relationship with UV also helped explain our contradictory findings about soil moisture. So remember, our expectations were that the pink flowers would be favored in drought conditions. And that's been shown in other studies where if you have a pigmented morph, it's usually responding to a dry or drought condition. Yet we see an increase in soil moisture with an increase in pink morphs. And the increase in soil moisture makes sense because as we're in a summer rainfall region, the rains have started, so the soil is damper. However, it could be that UV is just more stressful to the plants, and the presence of anthocyanins in these water available habitats isn't really detrimental. So if they're weighing up their issues, they're more stressed out by UV than they are by water. So our last question was whether one morph was more fit than the other. And right now, it seems to be a draw. I'm not convinced one way or the other yet. So to compare fitness between the pink and white morphs, we had tagged individuals that we knew were pink and we knew were white, and we had gone back to them and collected seed, and we're confident that they didn't change. It wasn't like a white flower came up and then we missed it and a pink flower came up. We germinated the seeds in the greenhouse. They were watered twice a day and kept warm for a few weeks. And then we found that white flowers produced more seeds, but pink flowers had better germination. So it seemed plausible that the pink flowers might be better at germinating because they're later in the season. And so they might need to germinate faster to establish themselves before the winter sets in. From other work in the greenhouse, we know that seeds produced within the season will germinate immediately. So it is possible for them to kind of fall out of their capsule and start germination right away. Alternatively, it could be that the pink flowers, so if a flower is pigmented, particularly with something like anthocyanins, the floral temperature tends to increase relative to its unpigmented an unpigmented companion. And so this increase in floral temperature might facilitate faster seed development. And this is something we'd like to test in the future. So hopefully this field season, we'll have a few temperature probes that we can stick into our flowers and find out for certain whether or not our pink flowers are indeed warmer than our white flowers. Of course, the question I always get about flower color variation in Rhodohypoxis is, what about pollinators? So I'm not a pollination biologist, or I'm not very good at this. And so our field observations have been somewhat haphazard over the year. So far, we've seen 3 different beetles, 1 very small brown moth that got away from us, and 2 bee flies that also got away from us. So I'm not really able to give more identification than that. Having a few lab members independently identify that the flowers have a scent has been really eye-opening. I don't really smell a lot of things, and this is not COVID related. This is just totally, I don't smell very faint scents. And that scent seems to be stronger at night, and the flowers are in fact open at night. So thanks to the night owls in our lab, we now have some night observations of our flowers. And so we plan to do some pollinator observations over a few different time points through the night at different populations this upcoming field season. So a master's student in the lab, Bianca Ferreira, is taking this on. And so we want to be able to definitively answer that question and figure out, are these things actually visited by anything? Originally, I didn't really think that pollinators were going to play a large role in flower color polymorphisms or variation, because if you look at the flowers, they are really set up well for selfing. And so the bottom picture is the inside of a Bowerii flower. The stigma is super, super small. It's inside. You have to really work to get into it. The anthers open right onto the stigma. So they just seem primed for selfing. Interestingly, crossing experiments in the lab have suggested that Bowerii and pink flowers of confecta and white flowers of confecta, I kind of use those interchangeably because Bowerii are predominantly pink run. So Bowerii and confecta, they're not necessarily super good at selfing. And then they are good at vegetatively reproducing. And so here we do find that our plants are vegetatively reproducing. Bianca has also, in her exploration to reproductive ecology, has dug up a lot of our plants and has found that there are little babies coming off of the sides. And so on the side picture, you can actually see the 2 babies coming off of a single adult. And so we do know that there is vegetative reproduction. So given the propensity for selfing to occur and clonal reproduction to occur, I'm not sure how strong selection pressures from pollinators will be, but we hope to have a more quantifiable answer at the end of this flowering season. And so I always like to end with what we know and what we don't know, and the list of what we know is much shorter than what we don't know as more questions were raised throughout our journey. And so what we found is that color variability seems to evolve multiple times in hypoxids or within the Hypoxidaceae family, and that there is a variety of mechanisms used to construct color. So it's not just anthocyanins or it's not just structural color. The species are using lots of different mechanisms. Then I'm relatively convinced that UV seems to govern the shift in morphs in some of our populations. And that I think UV likely has a larger role in color variability at a broader scale. And so what we still don't know is who pollinates these flowers. And hopefully we will find that out in the next field season or so. Then we're also wondering if UV contributes to color variability at a larger scale. Color variability is centered in South Africa. And so why is everything more why are the colors more variable here versus elsewhere? Then we wanted to know if there a link with reproductive ecology, flower color, and polyploidy. This I'm very excited about because we have some insight into the ploidy levels of Platypetala and Bowerii and Confecta, and how this might affect the anthocyanin biosynthesis pathway, and reproductive ecology in terms of vegetative reproduction versus sexual reproduction might give one morph the edge over another. So we're trying to figure that out. And then we want to know what's up with the tie-dyed flowers. We see flowers like in the middle of your slide here, where you have some white and some pink. We haven't observed them changing, so it's not, so at first we were like, oh gosh, this is like, Hilliard was right, they are going from white to pink, but then if you follow the same flower through its lifespan, it's just tie-dyed. So there's been some really cool work in Aquilegia where they show that there is some virus affecting the anthocyanin pathway, and so maybe there's something going on there. I don't know if it's viruses in rhodohypoxis, maybe it's something totally different, but it has gotten our brain cells moving. And so the path that lies ahead is relatively windy. We're moving forward with our reproductive ecology. And then when Steph and Henny were out in the fields last year collecting Poridias, they noticed this awesome diversity in Capensis, where there were monomorphic versus polymorphic populations. And we're really keen to learn more about the genetic diversity and the population genetics of those, and whether or not that links to flower color variability. In our abundance of time spent with the flowers, we've noticed that there's a lot of variability within a species on floral shape. And so the bottom left pictures are all Confecta flowers from the greenhouse. So we are quantifying the color, the shape of the flowers, just to see how much variability is within and between varieties. And then of course, I love hybrids, and we know that rhodohypoxis can reproduce with hypoxis and generate these beautiful peach colored hybrids. I have yet to see one in nature. This was apparently collected from a clear, and I've tried to find where it could be, but haven't since. So we might try and make some in the lab and see what we go from there. So with that, none of this work would have been possible or that much fun without the collaboration of the Peas Lab, whether they work specifically on rhodohypoxis or not. Further, my colleagues at BITS, Sabna Matura for our GC mass spec data, and Dr. Sandy Steenhuisen and Dr. Nick Macher at Kwakwa for their invaluable field assistance, insight, and generally just lots of fun on the mountain. And lastly, to the staff at the Vitsisuk Mountain Lodge for putting up with us for multiple days every year. And thanks very much for listening, and I look forward to hearing your questions.
Well, thank you, Kelsey. Gosh, that's That's pretty mind-blowing stuff you've got going there.
Oh, thanks.
I mean, I've heard bits of it over the months and years, but putting it all together, that was great. Thank you.
Thanks.
Excellent. Right, does anyone have any burning questions to start us off with? If you could just raise your hand. Okay, well, I'll start us off then while everyone thinks about it.
Thanks.
So I have a couple of questions, but probably with your last few slides, I was wondering, so do you think that there is a genetic basis where an allele actually gets switched on and off by this UV stimulant or whatever it is?
That's my working hypothesis right now. So we have seen in other plant systems that they can respond to environmental cues. And so particularly something like hydrangea, where you have the blue and the pink depending on the soil pH. And interestingly, malvidin is one of the pigments that can be affected by pH. So I hadn't previously considered soil pH as a variable that would be influencing this, just because the flowers are so close together. I couldn't imagine that pH would change in like millimeters of space. But maybe there's something that happens over the season and individuals are responding in their own time. Like maybe some are better at responding than others. So yeah, I do kind of think that the confecta is responding to the environment. And that's one of the reasons we don't think it's an example of color polymorphism, because it's not a single individual that's pink, it's an individual that puts up different color flowers. So, yeah.
Okay, good, thank you. Okay, I see Anina has a hand up. Please, you're welcome to switch your videos on when you're asking your questions. It would be nice to see faces and great. Anina.
Hi, Kelsey, thank you so much for the interesting talk. I was just wondering, in your reflectance spectrum of the flower colors, why did you cut it off at 350 and not 300 nanometers?
I think that was just for the visualization. We didn't actually see, there was like a little bump at the end there, but it wasn't anything spectacular. So, and it wasn't very different between the morphs. It generally had the same trend, but mostly for size.
Yeah, okay, interesting, thank you.
All right, anyone else? Anthony, please go for it.
Hi, thank you. Hi, Kelsey, that was a really brilliant talk. Thank you. It was very, very insightful.
Thank you.
My question, which I'm still trying to sort of bring together. Basically, I wanted to ask you, so in some of the, I've read in a few of the plant families, such as Polygalaceae being one example. In those families, it's known that a lot of their dark pinkish color, their deep pink color, comes from the concentration of volatiles that accumulate in the subdermis layers. And those volatiles, with a lot of the plants in that Polygalaceae family, they use those volatiles, they emitted at low intensities just above the petal surface to discourage the growth of fungi, like parasitic fungi and things like that. So do you think there could be a similar interplay with your rhodohypoxis flowers, like between how moist the soil is and like an interplay between that and how much volatiles the plant is able to manufacture, and at what maybe UV plays a role in how those volatiles evaporate and atomize and everything. So do you think there's a bit of an interplay there that might not have been explored yet?
if rhodohypoxis has taught me anything, it's to not rule anything out. So it's entirely possible that that's happening in the populations. And I'd be very keen to figure out if that might be happening or not. So yeah, thanks for that.
Great.
Good. Thanks, Anthony. Anybody else? So Kelsey, another thing that I was thinking of as you were talking of, Rhodohypoxis barii is generally found at lower altitudes, correct?
If you look at where they are relative to platypetala, they tend to be slightly at the higher edge. They're not as high as confecta, but they do tend to be slightly more elevated than platypetala if you look at the variety as the whole.
And they're also more in the Southernberg than central and northern.
Yeah.
So that would be colder. And so maybe temperature does have something to do with it, because they're certainly not getting the UV exposure that confect is getting up at the top there.
Yeah.
But they're a darker pink.
Yeah. So we're wondering if there is something different about the Southern Drakensberg populations that confecta wouldn't be experiencing. And it's entirely possible that it's temperature. We've noticed the relationship with soil moisture now, but we want to figure out more about how temperature could play a role in this too, particularly with the floral temperature itself. Like if the air around it is warmer and the flower itself could be warmer, what does that mean for reproduction? So yeah.
And habitat could also play a role, because I mean, if platypetalas on the rocky outcrops, for example, predominantly, then it's less damp and it's probably therefore warmer. Yeah.
My current hypothesis that I want to run with is the ploidy level variation in platypetala. So I think that I'm hoping that there's some duplicate gene copy interactions there that highlight why it's shut off in those plants and not others, and that the light pink might be the diploids. So we'll try and figure that out in the next couple of years.
Okay. Wow. Well, I'm glad you, the geneticist, and you know how to do that. Anyone else? No?
I saw a comment that the volatiles might be responsible for the slight fragrance in the flowers, and that's definitely possible. So it's certainly something we should think about and do some scent analyses with these guys to see if it changes or not. It'd be a lot of fun.
Okay. I mean, I was thinking about, you were talking about this being a more widespread thing at the top of mountains, and I was trying to think about helichrysum and the color ratios, because there might also be a predominance of pinker flowers up top there, but I'd have to go and look at that.
Yeah, you should. That would be really interesting. And it tends to be more pigmented. So it may not necessarily be pink, but maybe like red or like purplish. So
then Kenneth's asking a question there. Do you have any of the white to pink flowers in your collections? It would be interesting to play around with that. Kenneth, do you want to elaborate? I mean, or do you understand, Kelsey?
I think I understand where we have an individual plant that puts up a white flower first and then later on puts up a pink flower. Interestingly, we haven't noticed that yet with our confecta. We recorded what color we collected in the field, and we've noticed that they are all coming up very uniformly, like white or pale pink. So we haven't gotten a lot of the bright pinks in the greenhouse yet, and I'm not really sure, like if it is UV, it's probably because we're not like getting
under the sky.
Yeah. So but yeah, it would be really great to to get some of them to see if we can trigger that in the greenhouse for sure.
Great. Okay, good. All right. Any more ideas, suggestions, questions?
Hi, Glennon, sorry, may I ask another question?
Yes, please do. And then I see Anina's also got her
Great.
hand up again. Great stuff. Please do.
What I wanted to, perhaps it's not a question as such, but more just wanting to confirm or debunk something. So I wanted to just sorry. Yeah, so Kelsey, I wanted to just ask you, so with the findings you've got, that UV radiation is most likely what's influencing the color morphs in your rhodohypoxis at higher altitudes. Would that only be like when you say the mechanism of how it works is the higher the altitude you get, the more pink the flowers become? Is that something that only pertains to rhodohypoxis, to that genus, or maybe to the Hypoxidae family? Is it something that only pertains to that, or do you think it extends to other families? Because just an interesting sort of anecdote that I've found with species of Hesperantha, which is a different family, that's Iridaceae. I've found with Hesperantha bauerii, I've found that most of the pink flowers I found are actually at lower altitudes and the white ones are at higher altitudes. So I was just wondering what your thoughts are on this.
Yeah, so that's one of the things that we have been grappling with a bit, because what we tend to see suggests that we would have expected to find more white morphs at high altitudes. And if you go to these populations, the confecta is there's a lot of white flowers, and when we go, there's literally hundreds of thousands of flowers. So what we're sampling is a small percentage of what's there, and you can see that there is a change over the time, but it's not like your population is going from entirely white to entirely pink. It's going from mostly white to mostly pink, and that pink ranges from a light pink to a dark pink. So I do think that there's still some trend with the high altitude for confecta being more white than pink, but we do see this shift, and it's the shift in the morph frequency that's been really interesting to me. Like, why has what's driving this change? Because you could easily have confecta have all the colors out all the time, but that's not necessarily what you see. And at some of the populations, like at Sentinel, if you go up to the top of the peak, there's other rhodohypoxis species up there that are all pink. And then they might have a single white individual here or there. And it's the same at Sawney Top. Confecta co-occurs with rubella up there, and the rubella are all pink, whereas the confecta are in that range of white to pink, depending on the time of year. So it's possible that there is another variable that we haven't quite gotten a handle on yet. So yeah, there's lots to do here.
Now, I'm sure it's that combination of water and UV and probably other things, but at least those two, because that very wet year you had a lot more pink coming up.
That surprised us. We very quickly went back and looked at rainfall records.
Very interesting. Thank you, Kelsey.
Yeah. Thanks, Anthony.
Great. Okay, Nina, you have another question or point.
Yes, thanks. I just was wondering whether there are also other plant species occurring in the same areas that are also white or pinkish that might support your theory about the abiotic factors. And then I was also just curious, do you know whether these subspecies can crossbreed, whether they're compatible?
Yes, so I can definitely answer the second one, and yes, they are absolutely cross-compatible. We have 4 years of crossing data right now that the 3 varieties reproduce. In the 2 color morphs of confecta also reproduce. And then when we've been out at Sentinel, we see mostly, I assume they're helichrysums. They're an aster, like a straw flower type plant in our quadrats, and they tend to be either pink or light pink or white. But I haven't noticed if they differ at all. So, because we only see like 1 or 2 of them relative to confecta.
Right. Cool. Interesting. Thank you.
Thanks.
Okay, who's this? Patlani says, theoretically it says that the color intensity of anthocyanins depends on pH, methylation, and acetylation of these pigments. Could the UV mediate the color change through these?
That's a really good question, and I am not sure. I would have to think more about that and read a bit more to see if UV would affect that. Yeah, that's a really good question.
Good. All right, well then I think we've probably exhausted questions and ideas here, unless anyone has a sudden flash of inspiration. But that was really interesting, Kelsey, and lovely to see such a range of experimentation over the years as one answer has led on to the next question.
Yeah, thanks, Glennon. Thanks again for inviting me to do this. I really appreciate it. It's been fun to share.
Thank you so much. Thanks a lot. Sure. And thank you everyone for joining us today. Lovely to have you all present. Despite the heavy load of teaching that's happening again, we've got a good audience, and thank you for that.
Yeah, and, thanks very much.
Yeah. Please, if just a reminder, if you want your SACNAS points and you want a certificate, please write your name in the chat, and then Elizabeth will follow up on that. Okay. Right. Thanks again, and take care, everyone. Right. Bye.
Bye-bye.